We communicate all the time, but have you ever wondered how protein in our cells talks with each other? We are here to figure it out with Charu. She's a Ph.D. student at the Novo Nordisk Foundation Center for Protein Research. Let's do that. Ciao, Charu. I'm fine. You?
I'm good.
Are you bringing us where you work?
Yes, let's go and see around.
So is this your bench, Charu?
Yes, this is my bench. As you can see, it's the biggest bench. And another cool thing is we have our own set of pipettes. And nobody can steal it from you because you have name on your pipettes.
Oh, that's nice.
Yes. Let's go and have a look on my cells and what I do generally. Let's go to tissue culture. I take out my 4 dishes where I seeded my cells yesterday. So whenever we set up any experiment, we always need to have a control dishes where we do not give any drug. So these are my control dishes. Let's 2 dishes where I give them the drug. Now we incubate them for 8 days. We keep checking the cells if they're growing okay, if there is no contamination, like every 2 days. And so it's been 8 days. I had a look on them. They are growing fine, and I think it's time to count them. So we'll just take them out. Let's check if the dishes are dried, and I think they are. So the next step is to stain the colonies. And after you stain them, and you simply then wash them with water.
Right.
Now, these are the dishes where I added my drug. And since these drugs are very toxic, the cells cannot cope up with it and hence do not grow as well as the cells which were not treated with drug. And now, we can see that there is an effect, right? That the cells are very sensitive towards this drug. But to have an actual number, which is the interesting part, to make a graph, right? So, this is a machine that helps you to count the cells. It is called Gel Count. Super amazing, super fast, easy to work with. So you have your dishes. These are the trays, so you put your dishes here. Yes, so here it says count and we have this number. So you can already see I have my 2 dishes which were not treated, and of course there are more numbers of colonies growing in them, and there are the 2 other dishes where I actually treated with the drug. And then you can see that this is completely full. That means the cells have grown all over the surface of the dish, and here they're growing but not as efficiently as in the other wells.
So I have some few burning questions for you, Charu. What did you show us in the lab?
So it is something that we do regularly in the lab, or at least I do. It is called survival assay. So we give a treatment to the cells and see how well they are growing when you treat them. So in this case, I am giving a drug to my cells and I'm seeing how well they are growing.
Okay. What was the drug that you are using in this experiment?
It is called sumo inhibitor. So it actually stops a process which is very important for 2 proteins to be able to communicate with each other. And since the proteins are not able to interact and hence they cannot do things efficiently, that's why cells are not growing well if you treat them with this drug. What is the mechanism? I don't know. I'm studying that.
So this is your research project?
Yes.
But I was wondering, we always think that drugs are something useful, good, but here they are killing cells.
It depends in what context you're using these drugs. So some drugs can increase the function of your protein, some drugs can decrease the functions. So this is a very generalized or standardized method. So we stop a function and see how cells are doing. If they're doing fine, that means the protein doesn't have much role to play. But if the cells are not growing well, when you stop the function of a particular protein, that means this protein is important for the cell growth.
Can I make a stupid question?
Sure.
What were these blue dots that you showed us in the lab?
So those were actually colonies.
Colonies? What are colonies?
So cells, if they're dividing over the time, they form these big colonies of cells. It's like just cells making a society.
Okay.
It's like that.
Okay, so it's a lot of cells together.
Yes, so that we can see them when you stain them. Otherwise, if it would be just one cell, it's not visible. That's why.
What are you gonna tell us tonight?
So I'm gonna talk about how proteins in a cell communicate with each other and how one protein sends the message to other proteins in a cell.
That sounds cool. Looking forward for that. See you tonight.
See you.